So today's presentation, we're going to be talking about hybrid quantum classical graph generative models to target Alzheimer's disease. So I'm Linda.
I'm Lena.
And Narasimhan. Are you here?
I'm Narasimhan.
Okay, so starting out with a brief introduction to our project. So our project centers around the issue of drug discovery, which can cost billions of dollars and be very expensive, both in terms of money and in terms of how long it takes. So it can take up to 5 to 10 years just to generate one drug. And this is using classical methods. So a lot of times computational methods are used to speed up the research in recent years. And one thing that's been especially helpful in this is generative models of AI, such as GANs, which are generative adversarial networks. And we'll get more into those in a little bit. But basically what we're trying to do with our project is come up with a GAN type method to generate drugs that can target Alzheimer's disease in specific. So Alzheimer's disease is a progressive neurodegenerative disease, and it currently has no cure, which is why it's important that we're looking for drugs to target it. And it's currently one of the most common causes of dementia, which is also known as memory loss. And if you look over here on the right side, here's some diagrams that explain like drug discovery, and then the structure of a GAN, which we'll get into, and then some other models that we have as well.
So in preparation for building our model, we looked at a lot of different types of machine learning models. And in the end, we settled on GANs, which are Generative Adversarial Networks. So essentially how a GAN works is it's unsupervised machine learning algorithm, and it has two parts. The two parts is the generator and the discriminator. The generator will essentially generate the best possible fake data, and the discriminator will discern this between fake data and real data. So essentially what you do is that you put in a data set, and the generator will make the fake data, and the discriminator will use a data set to cut down the fake data made so that it is the result which you want it to be. So in our case, we would be training our discriminator so that it would cut down on the data so it can specifically apply to Alzheimer's disease. So another thing that's specific to GANs that we can use in our project is the latent QGAN. So this is a form of generative adversarial networks where images are mapped into a latent space with smaller dimensions. Essentially, the model creates its own latent space, and it uses that space to map out the data. The reason why this is helpful for our project is because to create a cure for Alzheimer's, we're looking at very small molecules. So to properly map out these molecules, we need to have a smaller dimensions and space so that the calculations are the most accurate that it can be. So this latent QGAN is good for visualizing the smaller objects like the molecules, and it also has higher quality data for drug discovery especially.
Okay, so given our previous work on generating molecules, we would now like to focus on generating molecules to target diseases, specifically Alzheimer's. Alzheimer's is a neurodegenerative disease in which a person may face symptoms such as memory loss, concentration issues, mood swings, aggression, and a general decline in motor skills at late stages. Alzheimer's is thought to be caused by a buildup of amyloid beta, and the buildup of this peptide causes a disruption in cell communication, which in turn produces an immune response. And this immune response is actually what causes the death of brain cells. Okay, so to target amyloid beta, what we want to do is basically plug in a database of inhibitors to amyloid beta and use the model to generate possible candidates. So we would use the discriminator to generate fake data based on the database of amyloid beta inhibitors, and based on that data, it'll generate new molecules which could actually also inhibit amyloid beta.
Okay, so it's worth noting that the task of targeting Alzheimer's disease is complex and multifaceted, and one of the other facets of it is the blood-brain barrier. So basically, for a drug to be able to affect Alzheimer's disease or amyloid beta at all, it needs to be able to pass through something that's called the blood-brain barrier. So the blood-brain barrier is consisted of a bunch of layers, including parasites, which are specialized cells that are associated with the brain's small vessels, and they help regulate endothelial response. And then there's also astrocytic end feet, which regulate water and potassium ions, and then regulate the metabolism, active ion transport, and transcytosis. And then there's brain endothelial cells, which are tight junctions, and they have a lack of pores. Tight junctions in circle cells, they have high electrical resistance. They're formed by plasma membrane proteins, claudin, oculin, and other GANs. And then they work with the adherence junctions, which are formed by catenins and cathephrins. Okay, so for a molecule to be permeable through the blood-brain barrier, it needs to have certain different molecular descriptors as listed over here. Being permeable is super important in determining the drug effectiveness, because obviously if it can't even reach the thing that's supposed to be targeting, it won't be able to do anything at all. So if you look at this chart here, there's some different values that show like relatively how important each of these molecular descriptors is.
So one molecule with potential to inhibit the amyloid beta is the SMART antibody. Before the development of the antibody, mouse monoclonal antibodies were employed to reduce the excessive levels of amyloid beta and tau proteins found in the brains of those plagued with Alzheimer's. However, there was no cognitive improvement, most likely due to the fact that memory deficit was related to damage done before Alzheimer's symptoms were observed, such as neuroinflammation or mitochondrial and lysosomal dysfunction. Therefore, a new heavy chain antibody called the SMART molecule was developed to target amyloid beta. It was injected into transgenic mice with Alzheimer's disease once weekly, and it was shown to have cleared amyloid plaque by about 60% compared to the untreated group of the mice with Alzheimer's disease. So this is an example of a molecule which it can be in the database of multiple molecules that target amyloid beta. So this is the kind of molecules we would be basing our model off of. So the molecules we generate could be similar to this and also have a high chance of being inhibitors to amyloid beta. So we talked about the structure of the blood-brain barrier a bit before, but we need to go more in depth to specifically how the molecule will get through it. The structure of the blood-brain barrier is very important to consider when addressing the challenge of optimizing a molecule to get through it. So there are three barriers: the microvascular endothelial cells, which make up most of the barrier, the blood and cerebrospinal fluid barrier, and the arachnoid membrane. It is also important to note how the cells are arranged. Tight gap and adherence junctions all play a huge role in how molecules are able to get through the blood-brain barrier. Tight junctions, which contribute to the blood-brain barrier selectivity, are especially important. So what impacts the permeability of a molecule through the blood-brain barrier? Size, charge, structure, hydrophobicity, and affinity of a molecule are especially effectual, as large and hydrophilic molecules are restricted, but low affinity molecules perform better than those with high affinity. So these are the kind of aspects that we consider while building our model, because the blood-brain barrier has a lot of different characteristics that you need to have for the molecule to actually pass through, and some that you don't. Because it's so hard to optimize a molecule to get through the blood-brain barrier, mechanisms of antibody passage can be useful. For example, one of these is the adsorptive-mediated transcytosis, AMT, which relies on electrostatic interaction and catenization to enhance uptake into the brain. Another is the carrier-mediated transport, CMT, which uses specific carriers for molecules. And another is receptor-mediated transcytosis, which uses receptors for antibody transport. There are also structural modifications to possibly make to antibodies, such as minimizing the number of hydrogen bonds, glycosylation, which is a carbohydrate attachment to another molecule, using natural carriers, and perhaps the cationization of the molecule. So,
Right.
so while there's different ways to get to the blood-brain barrier that isn't just the natural way. There's also, you need to take into consideration the different aspects of the molecule. So in our model, we plan to target both, as we will recognize that once we make the molecule with the certain specifics, that we also have to show that this is how it would be actually put into the brain.
Okay, so one thing that's worth noting is that our model actually is not complete yet, but we're just going to be talking about the basis of our model and what our general plan for it is. So the basis of our model is a model that we found called 10GAN, which is created by other researchers. And it basically uses transformer encoders, that's what 10 stands for, as the generator and the discriminator in the GAN, as we talked about previously. It uses various other techniques, such as a Monte Carlo search, as you can see here, and then reinforcement learning, and also masked multi-tad attention. And then the modifications that we'll be making to this architecture will be many of the things that we talked about on the previous slides. So in short, the blood-brain barrier permeability tests, and then the tests for amyloid beta inhibitors, and including like the data set for amyloid beta inhibitors, and then also incorporating quantum optimization, which we didn't talk about as much, but is something that we're currently working on.
Now we want to talk about the Guacamole benchmarking system, and this system is not a model. It's just for benchmarking, and we plan to use it on top of our main model. Guacamole is a system of benchmarking which allows models' performance to be compared to each other. It has two benchmarking systems, one for models generating molecules from a data set, and one for generating molecules with specific chemical properties. The Frechet ChemNet is a measure of structural and chemical similarity to a ChemNet database. The Kale divergence benchmark compares the difference between the current and previous generated molecules. Guacamole uses two systems to automatically remove molecules that are unstable or hard to synthesize. And the HTS method, which is extremely computationally expensive, and the Walter-Arty filters, which filter out molecules based on their structure.
For future research, we plan to explore more machine learning techniques and incorporate them into our models. Like we said before, we plan to use a generative adversarial networks, but there's also other additions we can add to the model, which we plan to explore. We also want to run on real quantum hardware, which is inspiration from the IEEE International Quantum Conference and reviewers. They encourage us to, instead of running on a quantum simulator, to run on actual quantum hardware. However, we are currently facing issues with funding on the quantum hardware.
Okay, so now we'll start acknowledgements. So we'd like to thank Dr. McMahon for his guidance throughout this project, and Neil McMahon for helping us deal with server-related problems. And we'd like to thank Dr. New for his guidance on resources that we could utilize during this project, and ASDRP for giving us the opportunity to conduct this high-level research, the XQGAN and QNetGAN group members, and the quantum chemistry group for being amazing collaborators. And to the right, you can find the QR codes for the references for this project, the JEI paper, and the IEEE conference proceeding. And thank you everyone for listening, and now we'd like to open the floor for questions.